So good morning students. Today we will talk about in the last class we had talked about the developing by plant breeding methods or plant breeding technique developing the disease resistance plants. Okay. After that we had also discussed how to develop that disease resistance plants, that is by the conventional method. Okay. After that conventional method, I had given some of the examples. Except that conventional method, there is also one more method, that is the mutational. Okay, or else we can also say that is the mutational breeding. So what is that mutational breeding? That's here. What are the examples developed by this mutational breeding for what? For developing the disease resistance plants. So see here, that is the mutation breeding. Mutation breeding. What do you mean by this mutation? First we will see here. It is a process by which it is a process by which genetic variations are created through changes in base sequences within the genes. So this is the definition of what? Mutation. See here, whatever the living organism will be there, they possesses the cells. Their cell has a particular, either that cell may be prokaryotic or eukaryotic, their cell possesses some genetic material. Okay. See here, if this is the cell at the center, see plant cell, we are talking about the plants. So the plant cell is the one of the eukaryotic cell. So in the cell, there is a presence of nucleus which is mounted by the nuclear membrane, and inside this nucleus, see this is the nucleus. In the nucleus, there will be the presence of chromosomes. The structure of chromosomes will be like this. Okay, in these chromosomes, there will be presence of what? There will be presence of certain genes. Within these genes, there is a presence of DNA. Okay, how the DNA will be there means, the DNA will be double helix structure. Okay, the DNA will be a double helical structure which is coiled with one another. Okay, so here the strands of DNA, it is made up of a or whatever the strand is there, it is made up of what? Nucleotides. What it is made up of? It is made up of nucleotides. Nucleotides means what? Means there will be presence of base pairs. The nucleotides such as adenine, guanine, cytosine and adenine, thymine, cytosine and guanine. These are the nucleotides. A, T, C, G. Always A pairs with thymine, C pairs with cytosine pairs with the guanine. You see here, adenine pairs with the thymine and cytosine pairs with the guanine. Okay. So this is about what? This is about these all are what? These all are nucleotides. Okay, nucleotides means that is the presence of nitrogen bases, the sugar and the phosphate. These three things are going to form the nucleotides. So whatever the DNA is there, deoxyribonucleic acid that it is made up of some of the nitrogen bases, they pairs with one another. These are the base pairs of the DNA. See here, adenine always pairs with thymine, cytosine pairs with guanine. So here, whatever the DNA structure is there, in this DNA structure, I will show you here. In this DNA structure, for example, this is the DNA double helix structure. Here, adenine pairs with thymine, cytosine pairs with guanine. In this way, there will be pairing is going to take place. Okay, well, the DNA duplicates means the DNA are going to form the copies. Okay, the duplicate copies of its own. See, how means in this way they are going to form the copies like this. So here, whatever the base pairs are there, in this way, they also gets duplicated. The base pairs are also going to duplicate it as the original DNA. This is the original one, and this is the daughter DNA or the duplicated DNA. In this way, their base sequences or base pairs are also going to duplicate it. What happens during the mutation means, listen here, it is a process by which there is a genetic variation, there is a genetic changes at the genetic level. Some changes are going to occur. Okay, some changes are going to occur which is at the genetic level. Genetic level means that is in the DNA, RNA, and the protein which is going to code for a specific trait. Okay, so that is related with a genetic material. So in that genetic material, if there is a change in the genes or the genetic material, then there will be change in the particular trait or a character of a particular plant. So see here, genetic variation are created through the changes. How that genes are going to change, the genetic variations are going to change means if there is a change in the base sequence, which are that base, that is the nitrogen base, what I had told you here. See here, these are the nitrogen bases. If some changes are going to occur in these base sequences, changes in the base sequences within the genes. I had told you these genes are in these genes there will be presence of DNA. In that DNA there will be presence of nitrogen bases. So is the adenine pairs with thymine, cytosine pairs with guanine. If something is going to change, if sometimes mutation error what we say, if adenine pairs with guanine, so this is the change. This is not a correct base sequence, not a correct pair. Otherwise, we can see if there is a long strand of DNA. Here, this is a strand of DNA. In this strand, if this is the C, then A, T, C, G, A. If this is a particular sequence, base sequence, which is coding for, which is making a particular character in the plant, if some changes is going to occur, for example, see, in the place of C and A, if it is going to T and G, it occurs T and G, so this change, if there is a change which is going to affect the characters, Okay, which is going to form a different characters in the plant. Not only in the plant, in every cell there will be certain change if there is a change in the base sequence, and if like that changes or variations occur at the genetic level, that is due to the change in the base sequence, that process is known as the mutation. What it is known as? It is known as the mutation. So here, after the mutation breeding, if like this changes is going to occur, for example, you see here, this is the base sequence of two duplicated cells or DNA. If here something is going to change, A is going to pair with the G here in this DNA, then totally that character is going to disturb and there will be change in the particular trait. So see here, if like this changes is going to take place, that why this changes is going to, what are the causes for this changes means, let's see here. Causes for mutation breeding. Causes for mutation breeding. What are the causes means? See here, and this mutation, it's not going to occur naturally. Okay, keep in one mind, in your mind, that mutation is not going to occur naturally, but it is going to occur artificially. Okay, so what are the causes means? This causes is going to induce mutation, or these causes are induced artificially. How it is going to form or occur, that is artificially. What are that means? First thing, by some radiations. How it is going to occur means, by some radiation. If this is the plant and this plant is under the sunlight, okay, for example, the sunlight or whatever the light rays are there, that contains the UV radiation, then X-rays, gamma rays. So all these rays, especially the UV rays, ultraviolet rays, X-ray and gamma rays, are going to create some changes in what? In the plants or for the mutation. Okay, so these are some rays which are artificially for the cause of what? For the cause of mutation. The next, what are the radiation? X-rays, gamma rays, then UV rays. These are the causes for what? Mutation. Next, that is the by use of some chemicals. Use of chemicals. And what are the chemicals, sir? And that particular chemicals by which the mutation is going to occur, sir, that is known as the mutagens. What they are known as? They are known as the mutagens. Next, that is by some gases, especially by smoking, whatever the cigarette smoking will be there. By that, also there will be changes in the genes is going to occur. Okay, so these are the causes for what? For this mutation. Then next, here, this whatever the mutation is going to occur, and if mutation is going to take place, this mutation will result in creating a mutation will create a new character in the organism. What is the result of this mutation? See, I think you had studied about the central dogma. Central dogma, that is in the chapter molecular basis of inheritance. That is RNA gets converted into the DNA by replication and transcription. If there is an error is going to occur, okay, error means something is going to change in that process, means then a mutation is also going to occur. That is also one cause for what? Mutation. So what is the result for that particular mutation, means that it is going to result in the occurring, or it is going to form a new character in the organism, which is, or a character or trait which is not present in the parental type. That new character can be seen in the new generation. So this is about the mutation. So whatever, see, we are talking about the mutation breeding. Okay, see here, breeding by process of what? Mutation. This is also one method for developing the disease resistance plants. See here, that here by use of chemicals or radiation, by use of certain chemicals and radiations, whatever the mutation is going to occur, selecting and using the parent plant, selecting and using the plants that have the desirable character, that have the desirable character as source in breeding, as source in breeding, and this process is called as mutation breeding. What it is called as? It is called as the mutation breeding. See, till now we had studied about only what is the meaning of mutation. Next we will go for the what do you mean by the mutation breeding. See here they had explained what is the mutation breeding by the use of certain chemicals. See, first of all, by using the plant breeding technique, that is selecting the germplasm, evaluating, testing, releasing into the commercial fields, that all steps we had studied in the plant breeding process. So here what I am saying means that plant breeding process can be developed, okay, by two methods, that is the conventional and second is the mutational. In conventional, the same steps will be followed, but in mutation, what happens means by using some chemicals and radiations, they are inducing what? That is the mutation. They want the mutation has to occur. So see here, whatever they are selecting, first step is what they will do. They will expose that particular plant with some chemicals and some radiation. Second step, they will select that particular plant. Select and using the plant. See, for example, this plant has a high yield. This high yield trait, whatever the character is there, that is developed by what? By mutation. Okay, by changing some of the base sequence in the plant or in the genes of the plant, they have developed this plant. So what they will do means, in that plant also they will select the plant and they will use that plant. They have a desirable character. What is the character over here means? That is the high yield. So this character is source in the breeding, and this process is known as the mutational breeding. Some examples I will tell you in the mutational breeding. Let's see the examples. See here, what are the examples? What are the plants has been developed by what? By the mutation breeding in moonbeam. In moonbeam. Okay, you are familiar with the word moon. Okay, which is one of the pulse. Okay, pulses. So here, which is resistance to moonbeam, which is resistance to yellow mosaic virus. It is resistance to what? Yellow mosaic virus that is induced by the mutation. What are the symptoms? What are the things you can see when a plant is suffering from the yellow mosaic virus insert? And that is the powdery mildew. This is also one of the disease in what? In the moonbeam. That is, one is the yellow mosaic virus YMV, and one more, that is the powdery mildew, which were induced by mutations. This resistance to that particular virus and this disease, it is developed by what? Mutation breeding. One more example is there, that is resistance to yellow mosaic virus in bhindi. First we had talked about the in moonbeam. Next we will see about in bhindi. Okay, yellow mosaic virus that is in bhindi. And what is the scientific name of the bhindi means? Abel Moscus. Abel Moscus escalenthus. This is the scientific name of the bhindi. And here, so what they did for the resistance by mutation means what? Transferred from a wild species. This bhindi with resistance with yellow mosaic virus, this were transferred from a wild species and resulted in a new variety of bhindi, and they named that particular new variety of bhindi as Parbhani Kranti. What they named? This is very important question for 1 mark. Parbhani Kranti. This is a new variety of bhindi which was derived from the wild species by which method means that is by the mutation breeding. Listen here, that is a first example in the moonbeam. They had got the resistance to yellow mosaic virus by what? By the mutation breeding and powdery mildew, which are induced by the mutation. And the second example is a yellow mosaic virus in the bhindi. Here they had given the scientific name of the bhindi, that is the Abel Moscus escalenthus. Okay, so this is the scientific name of bhindi. These bhindi, when they had taken for the mutation breeding, they derived from where they had taken, so that is from the wild species of Bhindi and they develop a new variety. By what? By the mutation breeding. What is a new variety of that particular Bhindi? Means new variety of particular Bhindi, that is the Parbani Kranti. This is one of the important questions for 1 mark. By mutation breeding, which new variety is developed by mutation breeding in Bhindi? Means that is the Parbani Kranti. Okay, so this is about what? Mutation breeding. Next, see here, in the mutation breeding, whatever the examples they had given that involves the same species, not they are not taking from the different species and not they are going to cross. Whatever the crossing method is there, that is by the sexual hybridization they are going to do, or whatever the process of mutation breeding is there from between the target and the source plant, they will do by a process that is by the sexual hybridization. Next we will go for the next topic, that is the polyploidy. Now listen here, that is the next topic, polyploidy. Polyploidy. Okay. So see here, what do you mean by polyploidy? Here the term is related with the set of chromosome, many set of chromosome. That is known as the polyploidy. So see here, organism. We are talking about the plants. So organism, what we are taking, that is the plant. Organisms. And what is that organism? That is the plant. Okay, organism, that is the plant which contains more than 2 sets of chromosomes, more than 2 complete sets of chromosomes, 2 complete sets of chromosomes and they are known as the that particular process is known as the polyploidy. The organism which contains, see, in a normal organism, either it may be animal or a plant, there will be presence of 2 sets of chromosome. But more than that 2 sets of chromosome will be there, means that is known as the polyploidy. Listen here carefully. Organism or plant which contains more than 2. Okay, more than 2 means either it may be 3, 4, 5, 6, like that. More than 2. After the complete sets of chromosome, that is known as the polyploidy. So here, depending on the number of chromosome, we are telling more than 2. Okay, depending on the number of chromosomal sets, the individual are given different names. See here, depending on the number of chromosomal sets in the individual, they had given certain names, are given different names. And what are the different names means? They had given the different names. And what are the different names means? That either if one set of chromosome is there means that is known as the monoploidy. Two set of chromosomes are there means that is known as the diploidy or diploid. Three sets of chromosomes are known as the triploid. Four sets are there means what they are known as tetraploid. Okay, if five sets are there means pentaploid. Then six sets means that is the hexaploid. So like this, due to some mutations, due to some changes in the chromosomes, due to addition or deletion of the chromosomes, there will be certain names they had given for the particular individual. What are that? Monoploid means only one set, two means diploid, three means triploid, four sets means tetraploid, five means pentaploid, and six means hexaploid. So here they had given certain different types of polyploid. So let's see the different types of polyploid. Next, types of polyploid. Types of polyploid. Here in the types of polyploid, so three types of polyploid discussed. What is that means? First is autopolyploid. Second is allopolyploid. Third is allopolyploid. What are these? Autopolyploid, allopolyploid, and next is autoallopolyploid. Okay, see here. What do you mean by this autopolyploid, allopolyploid, and autoallopolyploid means here if numerical increase of the same genome, numerical increase of the same genome. Okay, for example, here they had representing the particular chromosomes with see example that is auto. They have taken the example as auto triploid. Triploid means three sets of chromosomes will be there. So that is they are represented as the AAA. Here that is with the AAA. Okay, see what they are telling, there is a numerical increase. Number of chromosomes are going to increase. Okay, that is the numerical increase of the same genome. Same genome means same chromosome. Okay, here A. There is an increase of A once, twice, and thrice. So that's why we have taken example as autotriploid. So here it is going to increase. How means if you see in the autotetraploid means this chromosome in autotriploid it increases 3 times. In tetraploid it will increase 4 times. A. Okay, this is an autopolyploid. Only the chromosome number is going to increase. That is known as the autopolyploid. So here what happens means it increases the plant organs. If like this chromosomes are going to present more than 2 means in the autopolyploid it results in the increase in the organs, increment in the plant organs. What is the cause for this? Increment in the plant organs. There will be more number of plant organs due to the change in the chromosome number. Next we will see about the second type, polyploidy. Next, that is polyploidy. Here, this is developed by hybridization. Polyploidy developed by hybridization. Developed by hybridization between two species. If there is an hybridization is going to take place, see how this polyploidy is going to develop himself. That is first thing, that is the hybridization between the two species and followed by the doubling of chromosome, which is followed by doubling of chromosomes. In the autopolyploid, there is an increase in the number of chromosomes. Here, what is the normal number of chromosomes? That number of chromosomes is going to double. Okay, doubling of chromosomes. So how means see here, that is if AB is the normal number, normal set of chromosome is the AB. How it is going to double means A, B, B. This is these number are going to double. Okay. Next, the third one is polyploidy. See what happens means it develops a new species. I told you here there is a developing by what? Hybridization between the two different species. Okay, if two different species are undergoing into the mating process, what happens means it develops a new varieties. It develops a new species, and the new species name is wheat Nicotiana tabacum. Example, that is in the wheat, and the new species they had given as the name as Nicotiana tabacum. Nicotiana tabacum. This is one example. Except this one more is there. One more variety they had developed, that is Raphano brassica. These examples are important for your need points. Raphano brassica. Then one more, triticum. These are the new varieties of what? Wheat, which is developed by what? By crossing the two different species, wheat and rye. Okay, they are not going to cross with wheat and wheat, but wheat and rye, crossing with wheat and rye, and they got the new varieties, that is the Nicotiana tabacum, Raphano brassica, and triticum. And last one, that is the polyploid. Let's see here. In the polyploid, this is one type, last type of polyploid. Here, one genome is in more than diploid state. One genome is in more than diploid state. More than diploid state. More than diploid state means already there will be presence of two sets of chromosomes. For example, AA. You can take an example. AB. Okay, AB is there, and this is the two sets of chromosomes. Okay, they are going to double. They are going to double means AA is there, their number is going to double, and BB is there, their number is going to double. Like this, more than diploid state. Okay, that is the polyploid. Example they had developed here, that is the Helianthus tuberose. What is the example? Helianthus tuberoses. This is developed in the potato. Okay, so this is about what? Polyploid. Okay, so this is about the three types of polyploids. That is the autopolyploid, allopolyploid, and auto-allopolyploid. Before that, we had discussed about the mutation breeding. In that, we had given the examples as moonbeams, which is disease resistance to yellow mosaic virus and powdery mildew. One more example I had given, that is the mutation breeding, which is going to take place in what we call bhindi. What is the new varieties generated or developed means, that is the Parvani Kranti. Okay, so this is about the mutation breeding and autopolyploid. In the next class, we will discuss about the plant breeding for developing resistance to insect pest. Okay, so we had discussed how the plants has been developed with the resistance to diseases. Next, with the insects pest, which is also responsible for a major crop loss or distraction of the crops. Next, after that, plant breeding for improved food quality. In that, we will study about the biofortification. Then that with a single cell protein, SCP. Okay, so thank you. If you have any doubt, you can ask in the chat box or in the WhatsApp group. Okay, thank you. Please note down the notes, whatever I am writing on the board. This is insufficient for you, the theory exams. Okay, thank you.